This video clip talks about the liver and gallbladder. Like the pancreas, liver is also a mixed exocrine and endocrine gland. The difference here is that these functions are performed by the same cell. In the endocrine portion, liver produces plasma proteins and lipoproteins that are released directly into the bloodstream. Bile is produced in the liver cells but transported out via duct system, and therefore it is referred to as exocrine system. This schematic describes the organizational structure of liver called classical lobule. The liver is composed of these units that act in concert to carry out its function. In the center of the lobule is the central vein, shown here by this arrow. This receives blood from the hepatic artery and vein, which is located on the outside here. While hepatic artery delivers 25% of blood, portal vein delivers 75%, as shown by the size of these two respectively here. However, the oxygen demand from the liver is equally shared by the artery and the vein. Hepatic artery and portal vein are located in the periphery of the lobule, and together with the bile duct, you can see this here in this green, form a characteristic feature called portal triad. Sometimes lymphatic vessels are also seen in this area, but the name stays the same, portal triad. In the next few slides, we will look at the finer structure of liver lobule, its cellular composition and arrangement and function. The schematic shows that hepatocytes are arranged in plates, and these are related to the blood supply in the sinusoid, which comes from the portal triad towards the central vein, so the blood flows from the outside in. In this picture, the hepatocytes are shown in green to highlight their arrangement in plate form. In this one here, the area that's been blanked out and appear white are the sinusoids. As you can see, they are right next to it, the hepatocytes. In this particular picture, you can see that the sinusoids are now filled with blood as represented by the red color. At high magnification, this becomes clearer. Here the sinusoids are shown in blue. You can see that here. And arrows pointing towards the central vein indicating blood flow from the portal triad into the central vein. Hepatocytes are shown as adjacent blocks with green arrows here, which indicates the flow of bile from the center of the bile duct to the center of the bile duct in the portal triad, which is located here. This is done through these small channels called canaliculi, and we will look at their structure later properly in the EM, here and in the lab. In this particular slide, you can also see the Kupffer cells shown here in yellow, and these are the cells referred to as the liver macrophages. In the lab, you will also study relationship between hepatocytes and the endothelial cells of the sinusoid. This EM shows a gap between these two cell types and is referred to as space of Dis. So this is one hepatocyte here with the nucleus, and here's another one here with the nucleus, with the bile canaliculus here between the three hepatocytes. But here, there are parts of the endothelial cells which are juxtaposition to the hepatocytes here. The space between these two cells is referred to as the space of Dis. It's named after Mr. Dis, and In order to understand the functional or pathological aspects of liver, it's important to know the organizational structure around the lobule. There are three concepts to master here. First, there's a classical lobule, which we spoke about earlier, which has the central vein in the middle of it, and the portal triads are on the periphery. This is considered the endocrine lobule because of its main function of excreting directly into the bloodstream that drains into the central vein here. So the blood is flowing from the outside to the inside. Concept number 2 is the portal lobule. In the portal lobule, the center is the portal triad as indicated by this black dot. And on the periphery are these central veins coming from the adjacent lobules. Once again, the drain is from the outside in, but in this case, the drainage is the bile that's traveling from the central vein towards the portal triad. The third concept is the liver sinus, which is also referred to as the metabolic lobule. It is related to the metabolic activity of liver and is commonly involved in pathologies. This is a zoomed-in view of that metabolic lobule, and it shows you how the pathologies may arise. Zone 1 area, here in purple, receives the highest level of oxygenated blood that gradually decreases as the blood flows towards zone 3 in the yellow here. Since zone 3 receives the lowest amount of oxygen, even under normal conditions, any disturbance in the blood oxygenation status would exert adverse events in this area. One of the main functions of the liver is detoxification. As such, zone 1, again in the purple here, is exposed to the highest level of toxins that are being eliminated as blood flows towards zone 3. In this instance, liver is exposed to a high level of toxin substance here in zone 1. So zone 1 is highly susceptible to damage from toxins. So liver biopsies are used to evaluate the microscopic location of tissue damage to assess particular pathologies. Like in other organs, liver has neighborhood where different cell types reside in to perform specific functions. This slide shows a typical neighborhood where hepatocytes meet sinusoid. Hepatocytes are active in synthesizing and secreting proteins as evidenced by prominent rough endoplasmic reticulum and mitochondria within their cytosol. Endothelial cells line the sinusoids, and you can see them here with red blood cells being in the sinusoid. And you can also see the Kupffer cells incorporating themselves into those sinusoids. This picture also shows the space of this in a more pronounced way, the space between the hepatocyte as well as the adjacent endothelial cells. Bile canaliculi can also be seen in the adjacent hepatocytes here, for example, which are connected to each other with strong zona occludens, which allow adherence of adjacent cells strongly. One other thing to note here, that there are some resident specialist cells highlighted here as ETO cells. These are cells which store vitamin A, remodel extracellular matrix, and produce growth factors and cytokines. And they also regulate the sinusoid lumen size in response to prostaglandins and thromboxanes. They are also considered stem cells that give rise to hepatocytes during tissue injury. So during that repair phase, overactivity of these cells can produce excessive amount of extracellular matrix and thus leading to fibrosis. Bile produced in the hepatocytes consists of two main constituents. The bile pigment, which is composed of bilirubin and biliverdin, is produced in the smooth ER by processing hemoglobin from senescent red blood cells. Bile salts that are essential for emulsification, solubilization, and transport of fat in the intestine. They carry out this function by having both hydrophobic and hydrophilic ends that act as detergents. These pictures emphasize the biliary system in the liver. The picture in the middle is a nice histological demonstration of bile canaliculi, along with the hepatocyte plate pointed by the arrowheads as indicated here, here, here. So these are the hepatocytes, individual hepatocytes, and the canaliculi running along the length as depicted in this picture here by the green arrows going to the bile duct here in the portal triad. And this picture here is a relationship picture between how the bile is transported from the liver into the gallbladder and eventually also released into the GI tract. Finally, the gallbladder. This is a thin-walled structure that is characterized by highly folded mucosa with simple columnar epithelium. Gallbladder can distend because of these foldings and in cross-section appear to have cystoid spaces, and we will see that in a histological picture a little bit later. So histologically, it also lacks conspicuous submucosa. Because the muscularis mucosa is also thin, lamina propria appears to be sitting directly on muscularis externa. Part of gallbladder is attached to the liver, and that part has adventitia on the outside. The remainder surface of the gallbladder is classified as serosa. The tall columnar epithelium of gallbladder concentrates bile by as much as 10 folds. This is accomplished by actively pumping of ions and water across the epithelial cell layer with absorption of fluid by lymphatic vessels and capillaries in the lamina propria. This is a histological view that summarizes the features we discussed of the gallbladder. Note the extensive infolding of the mucosa, which appear to have these cysts, but matter of fact, these are the foldings which have been cut in a plane of section that allows the view of the lumen of the gallbladder to show up in these foldings here. You also notice there is lack of that submucosa. There is no real separation between mucosa and muscularis externa. The muscle layers in the muscularis externa are also running in multiple directions for the whole gallbladder to distend in all directions. One other thing to note in this particular slide is the adventitia rather than serosa. So you can see this muscularis externa ending here, and then all this is the adventitia. This concludes this video. Further information is available in lecture notes and histology lab guide and pre-lab. Thank you.